So now we're going to transition over into our first invited session of the event. And so I think this is a great follow-up to Ben's wonderful talk, really motivating the framework, as the first session is going to be centered around vertical data science and medicine. And so I think Ben's work and the following session is really going to showcase the need for reproducible, reliable data analysis within areas like medicine, where the decisions being made are having this direct impact on people, right? And I think the motivating examples you're going to see definitely come across that way. And so first off, our speaker is going to be Tiffany Tang, who is a postdoctoral researcher with Ji Zhu and Liza Lavina in the University of Michigan Statistics Department. Her research interests are primarily problem-driven and lie broadly at the intersection of statistics, data science, and medicine. She will be joining the University of Notre Dame as an assistant professor in fall 2024. Congrats, Tiffany. And she previously received her PhD in statistics from UC Berkeley. I think we can all guess we were part of the BIN lab for sure, as she's been mentioning throughout that wonderful talk, where she was advised by BINU. And so thank you again to Tiffany, and I'll let you take it away. Oh, I should say your title. She's going to be speaking on low signal iterative random forests. Thank you.
All right, well, thank you so much, Anna, for the very kind introduction. I'm very excited to be here today to talk to you a little bit more about a problem that Ben has kindly foreshadowed for me, our BioHub collaboration on low signal iterative random forests. And since you've kind of seen a little bit of this already, I'm going to go through this pretty quickly, but generally our goal in this project is to develop an end-to-end pipeline for identifying both the genes and, more interestingly, the gene-gene interactions that affect a particular heart disease called cardiac left ventricular hypertrophy. And briefly, so if you have left ventricular hypertrophy, which is a pretty common heart disease, you have this thickened heart wall of your left ventricle, and having this thickened heart wall, it actually makes your heart have to work harder in order to pump blood out to the rest of your body. That extra stress over long periods of time over your life can lead to things like sudden cardiac death, arrhythmias, and heart failure. Okay, but what I'd like to emphasize here is that, well, we have this, we want to develop an end-to-end pipeline, which entails both this data-driven scientific recommendation system, where we hypothesize relevant genes and gene-gene interactions that we then translate into the wet lab for experimental validation. All right. And I'd love to spend, a whole talk talking about the experimental validation and all the cool science that comes out of it. But for today, and I know many of us are more on the computational side, I'll mostly be talking about the data-driven scientific recommendation part. Okay, and with our goal being to detect primarily these gene-gene interactions, or what is called epistasis in biology, there are several challenges that arise for this problem. The first being a purely computational one, in that, well, an exhaustive search for these gene-gene interactions can be computationally expensive and often prohibitive. So for example, if you have 100,000 genetic variants, which is well within the data that we see nowadays, and if we wanted to search for all possible pairwise interactions amongst those genetic variants, that's 100,000 choose 2 combinations, or 5 billion pairwise interactions that we would have to search over. And needless to say, that's computationally challenging, and we pretty much never have the sample size in order to conduct so many searches. And so this brings up the question of, well, okay, we need to make some assumptions and pose some sort of statistical model here, but what is an appropriate model and what are appropriate assumptions? Okay, so in particular, why restrict ourselves to just these pairwise interactions? What about higher order interactions that depend on 2, 3, or 4 or more variants? And also, what about nonlinear interactions? So these are all choices that we have to make in this process. But the final challenge, and maybe the kicker, as you will probably hear throughout the day, is real-world data is messy, noisy, and in our case, you will see incredibly low signal. Okay, and so with that, what is the data we are using for this problem? We are leveraging data from the UK Biobank, where here we have genetic information measured from 15 million features, so very ultra-high dimensional problem, measured in the form of SNPs, across 30,000 patients. And so if you're not familiar with SNPs, they're single nucleotide polymorphisms. You can just think of them as positions along your genome at which you frequently observe mutations in the population. And then this number in our data matrix is going to be the number of mutations you observe for that particular individual at that position. So it's just a matrix of zeros, ones, and twos. And we like to use this genetic information in order to learn something about our phenotype of interest, that being left ventricular hypertrophy. The challenge being its incredibly weak prediction signal. So for the longest time, maybe like 6 months or so, we were using the phenotype called hypertrophic cardiomyopathy. No matter what we threw at the data, no matter what prediction model, no matter what pipeline, we could never beat the 50% balanced classification accuracy, which is to say our models are doing no better than random guessing. So maybe that tells you a little bit about how difficult this problem is. But that also means, thinking about it, like how are we supposed to be able to trust this model enough, this model that may be no better than random guessing? How can we trust it enough to recommend these gene and gene-gene interactions for our collaborators who are then going to spend thousands of dollars and numerous hours just trying to validate this? So back to the drawing board, and there was quite a bit of work here and months of discussions on trying to come up with a phenotype still related to left ventricular hypertrophy, but has some kind of signal that we can learn from the data. And I'm going to sweep all of this under the rug and perhaps do it in injustice. But skipping to the end of the story, we ended up trying to predict what we call left ventricular mass, which is often used to quantify left ventricular hypertrophy. And this you can compute from cardiac MRIs using deep learning. Okay, but now running with this data, very noisy, messy, low signal, the question becomes, how can we learn about reliable gene-gene interactions? Okay, and so this brings me to our method called low signal iterative random forest, or LOSIRF, which essentially at a high level comes in 3 stages. So going through each one of these in turn. So first step is dimension reduction. We're going to have to do something about those 15 million SNPs. And we're going to borrow tools that are specific to the domain that have previously worked well. In our case, we're going to use a GWAS, a genome-wide association study, to reduce the number of SNPs of interest from 15 million to 1,400. So question that should be in your head is why 1,400? So here we used a prediction check to see, to kind of choose, like, are we going to use 500, 1,000, 2,000, so on and so forth. And ended up 1,400 and ended up being the best. Once we took, and running with these 1,400 SNPs for the remainder of the pipeline, we then actually binarized the LVM phenotype into 2 groups. So backing up just a bit, it turns out that if you actually just use the SNPs in order to predict the LVM phenotype, which is actually a continuous variable, the prediction accuracy still hovers around like a 0, like the R-squared being around 0. So we're not doing too much better than predicting just the mean. And so what we did is actually try to simplify the problem to make it easier for ourselves, by which I mean we take this continuous phenotype, LVM, take those with the highest LVM, group them into the high LVMI group, and those with the lowest LVM, group them into the low LVMI group, and then omit the middle portion and ask, can we predict those who have very high LVM, or that is actually those with large hearts from those with small hearts, in hopes making the problem easier for ourselves and in some sense boosting our low signal. But then there's the choice of, well, what is the threshold we are going to use? And so what we did is chose multiple different thresholds, 15, 20, 25%. And then for each of those thresholds, we ran our model, iterative random forest, to try to identify these higher order nonlinear interaction candidates. Okay, so just to recap, for each binarization threshold, we are fitting IRF, iterative random forest, which is developed by Ben, Carl, and Samantha, and Carl's here in the audience, using IRF to and using IRF on the GWAS filtered SNP data in order to predict our now binarized LVMI. Okay, and at a very high level, iterative random forest you can think of as just a stabilized version of random forest. So here, again, heavily guided by the PCS framework, IRF induces stability in IRF in order to improve its interpretability. And how does it do so? Well, it does so iteratively, as the name might suggest. So in the first iteration of IRF, we first train a normal random forest, where here, a random forest being a collection of decision trees. At each split in the decision tree, we have chosen some SNP feature to make the split upon. But what we're going to do is actually take the feature importances from that random forest and use these as weights in a subsequent iteration of random forest, where now we train a random forest using weights such that the features that were important in the previous iteration are upweighted in the next iteration, and then those that were unimportant in the previous iteration get downweighted. So we can do this many times. Generally 3 to 5 is enough. But what this will do is actually encourage the trees to split on more similar features and hopefully become more stable and easier to interpret. But also, hopefully, it does so without sacrificing too much in terms of prediction accuracy. So this you can check whether or not it's actually doing so using something like the out-of-bag prediction error. And it turns out that IRF actually performed better than all of the other machine learning methods that we tried, including just an ordinary random forest and some simple deep neural networks. And achieves a very humble, modest 55% classification accuracy for predicting the binarized LVM. So not great, but still better than 50% that we saw for months and months, and then better than some of these other machine learning methods that we've come to know. All right. And so what's the upshot of all of this by stabilizing the random forest? Is now we can actually leverage very efficient searches in order to identify sets of co-occurring features that commonly occur together along decision paths across the random forest. And these sets of commonly co-occurring features are what are the interactions that are recommended by IRF. And this has worked very well for many problems identifying interactions on transcription factor binding, even epistasis and red hair. But with everything, there are some limitations. In our case, IRF actually can only identify interactions that are stable enough in the random forest. But for our low signal problem, and also with having highly correlated SNP features, turns out that any of the SNPs of interactions are actually incredibly unstable. They only occur maybe once or twice across the random forest. So how are we supposed to determine whether or not it's one SNP interaction is better than another? It's very difficult. And so what we did is actually, rather than going for these SNP interactions, we went for gene-gene interactions, where here a gene is just a collection or group of SNPs that are nearby each other on a genome. Okay. But the second issue arises whenever we're trying to rank those gene-gene interactions. And so at a high level, like IRF ranks these interactions based upon how frequently or how stable they appear within the random forest. But there are some genes that only consist of maybe 1 or 2 SNPs, and other genes that consist of 100 SNPs. So how are we able to compare the frequencies of these two types of genes? Because longer genes will naturally be more frequent in the random forest. There's this bias towards larger genes. And so we need to develop a new feature importance score in order to rank the gene-gene interactions. And so I don't have too much time to go into what exactly our feature importance score is, but I just want to give you a very high level idea behind, or the driving idea behind this new feature importance score. And that is this paradigm shift between a global importance or global feature importance, where you measure the importance of each gene for the entire population, and then one that is a local feature importance, where you measure the importance of a gene per individual. Because the issue at the end of the day is, well, genes that are very small have low frequency of occurrence in a random forest just by random chance, and longer genes have a high frequency of occurrence, and so these are not comparable across genes. However, if we're able to obtain a local feature importance and measure the importance of each gene for each individual, now you can look to see, well, okay, you can make a comparison within a gene and look to see, well, is this gene being used differently for those with, say, high LVM versus those with low LVM? And then perform your favorite 2-sample test per gene to understand, well, whether or not there's actually a significant difference here. And so I'll leave out the details on how actually to compute each of these local feature importances due to time. But that wraps up our low signal iterative random forest pipeline. Is finally we rank the genes based upon this new importance score. And at the very end of the day, we obtained gene-gene interactions that were stable across all 3 binarization pipelines. In particular, we ran experiments on 2 of these gene-gene interactions, which were experimentally validated in high-throughput single-cell knockdown experiments, which was super exciting. And I'm running very low on time. So just wrapping up with the key takeaways. it was very exciting to see like these experimentally validated epistatic regulation of cardiac hypertrophy. Much of epistasis and the work there being done is relies solely on the data. So to see this actually hold up in experiments was a really exciting and rewarding experience. We have now this method, low signal iterative random forest, which can recommend these gene-gene interactions and tailored for low signal data. And then finally, last thing I would like to end with is this was only possible through collaboration and iterative cycle of feedback. And I have, this work certainly is not possible just by me alone. Everyone on this page has played an instrumental role in making this happen. In particular, shout outs to Bin and you and Ashley, who are the main PIs, and then Chunru Ron, my co-first author, and the many others who are here, including Anna and Carl. And with that, I'd like to thank you all for your time.
So Tiffany's actually very on time. So we can have, I think, at least 2 questions from the audience. And I believe we have volunteers on both sides to bring you a mic if you want to raise your hand.
Great talk. For the validation, I assume it's different. You can only do the knockout in the iPSC cell lines, and then it will be different to the clinical phenotype. So can you comment on that? And then people can also do systematic perturbation for many gene pairs, and then could you learn something help for the analysis of the germline genetic variants that,
Yeah, that's a really good question. So to recap, so for the experimental validation, so they created these iPSC cells derived from like human stem cells. So a whole bunch of work went into making sure that those cell lines are both standardized, relevant. That would, I think there may have maybe at least one, perhaps 2 or 3 papers on that, just to kind of confirm like the validity of those cell lines. And the second, is there?
Just if for those perturbations, you can do systematically many perturbations, and then quite often you can like mutate 2 genes in one cell, see the consequence. Would that help discovery?
Oh, right. So for the experimental validation, we ran several different kinds of experiments. One where we knocked down each gene of interest individually, marginally, and then we had another cell line where we knocked down both of the genes simultaneously. And then we ran some statistical analyses to see whether or not knocking down both of those genes is like interacts epistatically compared to just knocking down each one marginally. Right. And so we found 3 stable interactions from our pipeline and only ran experiments trying to validate 2 of them just as a pilot study. So yeah, great questions. Thank you.
Thank you.
So great talk. I just want to ask more about the iterative process that you mentioned. So you basically update specific features in the random forest in order to make them more similar, you said. So I'm not sure whether you could elaborate on that, what the purpose of the iterative process is.
Yeah, so the iteration from iterative random forest induces some stability into kind of the SNP features that are being used to construct the random forest, right? And so by doing so, we encourage the trees to become more stable and becomes easier to interpret without reducing the prediction accuracy. That's really important. We do still need that reality check and maintaining the high prediction accuracy. But if we are able to maintain that high prediction accuracy while simplifying the model, I'd say that's a big plus. And then with this more stable model, then you can now find pairs or triples of SNPs or genes that appear very frequently together along the random forest, and then dig deeper to see whether or not those are interesting interactions to then follow up via wet lab experiments.
And which features do you keep typically?
Oh, so this is a soft kind of weighting scheme. So all the features essentially have a chance to be picked up in the random forest, and you do this kind of this iterative process. Never or usually doesn't zero anything out completely.
Thank you.
One more question.
Hey, thanks for the talk. This is a really cool method. I have two questions. One is whether you see this being applied to also interactions of variants within a gene. And the other question is, out of the gene-gene interactions that you found, how they compare with existing pathways, or are you discovering new pathways, new protein-protein interactions?
Yeah, those are both great questions. So the first one, I think discovering interactions between SNPs within a gene is a very challenging problem. I think if you have a target gene like that you already know that you want to explore further, this would be possible. If you're starting with like say the 30,000 genes in the whole genome, like that, I think that might be difficult to get at in like one go. And I think maybe an iterative process would be better. But if you like have particular genes that you want to zoom into, then for sure, then you can certainly use IRF and a lot of these methods in order to identify these gene-gene interactions. And regarding the pathways, yeah, I think they are, so CCDC141, this gene that kind of popped up in all of our gene-gene interactions, is actually a very relatively understudied gene. So still a lot more work is to be done to understand like how this works. Or what role it plays in like existing pathways.
Let's thank our speaker again. Thank you, Tiffany.